Protein purification. The major aim of protein purification is to isolate a particular protein from a complex mixture. This protein is generally known as protein of interest. So multiple processes in a series are run to do so. The first step of protein purification is cell disruption. In cell disruption, different detergents are used to destroy the cell. Cell is being disrupted to take out the protein of interest along with other contents from it. Bacterial cells have a cell wall, so we use special enzymes called lysozymes to fragment the cell wall and detergents to break the cell. Now the cell debris can be removed by centrifugation. We employ centrifugation to remove cell debris from the solution. The supernatant contains proteins along with nucleic acids. To separate proteins from these nucleic acids, we use particular enzymes. These enzymes are called DNAs and RNAs. These enzymes gobble the DNA and RNA molecules. So the remaining mixture contains only proteins now. The proteins are then precipitated in a process called salting out. This process is mainly carried out by adding a salt like ammonium sulfate to the mixture. This process decreases the solubility of proteins in water by adding the salt. In a normal mixture, water molecules surround the hydrophilic part of proteins. These hydrophilic parts of protein contain hydrophilic amino acids, which interact with the surrounding water molecules. But when we start adding salt into the mixture, the salt ions compete with proteins to interact with water molecules. If we keep adding the salt, it weakens the interaction of proteins with the water molecules, but strengthens the protein-protein interaction. As a result, the protein molecules rush closer to each other and unite to become stable. The precipitated proteins are then removed from the mixture, and they are treated with different chromatography techniques for further purification.
That is it for today's video. Make sure you are subscribed to the Science Info channel. Like and share this video if you wanna help others. Stay tuned. I will see you in the next video.